Welcome to the most mind-bending question in all of biology. How does a single featureless cell, invisible to the naked eye, orchestrate the construction of a complete human being with a beating heart, a thinking brain, and trillions of perfectly organized cells? This is the central enigma of embryology. And here's what makes it so explosive for medicine. The cellular rules that govern human development don't just explain how babies are built. They explain cancer progression, they explain why your heart can fail, and they are the very foundation of the regenerative medicine revolution happening right now. Every pathway, DNA replication, positional signaling, tissue homeostasis, traces back to decisions made in those first extraordinary hours after fertilization. Embryology isn't just a subject for first-year students. It is the master code of human life. And once you understand it, you'll never look at disease the same way again.
Why does every serious medical curriculum force you to master embryology? Because it is the scaffolding under 4 massive clinical domains. Pillar 1, anatomical logic. Adult anatomy only makes sense when you trace its developmental origins. Why does the recurrent laryngeal nerve loop around the aorta? Because of aortic arch remodeling in the embryo. Without embryology, anatomy is just memorization. Pillar 2, developmental defects. Physical anomalies affect approximately 1 in 20 people when you account for late-presenting defects. That's a staggering clinical burden. Pillar 3, reproductive technology. IVF, ICSI, embryo grading, blastocyst transfer, none of these exist without embryological principles. And Pillar 4, adult disease origins. The fetal programming hypothesis tells us that gestational insults reshape cardiovascular risk, metabolic syndrome, and even cancer susceptibility decades later. Embryology is not background noise. It is frontline clinical science.
Here is a number that should stop every clinician cold. In Western Europe, approximately 2.4% of newborns carry a significant developmental defect detectable at birth. That sounds manageable, but that is only the tip of the iceberg. An additional 2.6% of individuals will develop defects that only become apparent later in childhood. Structural heart defects, sensory neural deafness, renal anomalies, conditions that were embryologically determined but clinically silent at birth. Add those together and you reach approximately 5% of the entire population. 1 in 20 people carrying a developmental defect rooted in early embryological errors. For exam purposes, remember, congenital heart defects are the single most common category, affecting roughly 1% of live births alone. This hidden iceberg of developmental pathology is why embryology isn't optional knowledge. It is the foundation of pediatrics, cardiology, genetics, and surgical planning.
Let's get to the core puzzle. You begin as one cell, a zygote measuring approximately 80 microns across. A typical human somatic cell is roughly 10 microns. That single oversized egg must transform into a complete human being through 3 sequential and precisely choreographed processes. First, multiplication. The zygote undergoes relentless mitotic division, scaling from 1 cell to the 37 trillion cells of an adult body. Second, differentiation. Those genetically identical cells must diverge into over 200 specialized cell types. Hepatocytes, cardiomyocytes, neurons, osteoblasts, each with a completely distinct structure and function. Third, deployment. Differentiated cells must physically migrate to exact anatomical coordinates and assemble into functional organs. If any one of these 3 programs fails, wrong timing, wrong location, wrong signal, the result is a congenital defect, a miscarriage, or, critically for your exams, a predisposition to malignancy.
Here is the paradox that unlocks all of developmental biology. Every single cell in your body, from your retinal photoreceptors to your pancreatic beta cells, contains the exact same DNA sequence, identical genome. Yet a muscle cell looks and behaves nothing like a neuron, which looks nothing like an osteocyte. If the DNA is the same, what makes them different? The answer is gene expression, not gene sequence. Differentiation is a software problem, not a hardware problem. The landmark proof came from Dolly the sheep, cloned in 1996 from a fully mature mammary gland cell. That adult cell's nucleus, when placed into an enucleated oocyte, reprogrammed completely and generated an entire organism, proving the full genome remained intact and totipotent. The signal environment surrounding a cell determines which genes are switched on and which are locked away. This is the foundation of epigenetics, and it explains why identical twins can have dramatically different disease profiles despite identical DNA.
Immediately after fertilization, the zygote enters a phase called cleavage, one of the most biologically unusual processes in human development. Here is what makes it extraordinary. The cells divide rapidly, but they do not grow between divisions. Each daughter cell, called a blastomere, is exactly half the size of its parent. The total volume of the embryo remains constant, essentially the same 80-micron diameter of the original egg. This rapid size reduction continues through the 2-cell, 4-cell, and 8-cell stages, until by the 16-cell stage, you have a compact ball of tiny cells called the morula, named from the Latin for mulberry, because that is precisely what it resembles histologically. This shrinking to normal size process is critical. It brings cells down to the nuclear to cytoplasmic ratio required for normal gene expression. Clinically, abnormal cleavage rates are a primary marker of poor embryo quality in IVF. Embryologists assess cleavage symmetry and timing to select the best embryos for transfer.
At approximately day 4 to 5 post-fertilization, fluid begins accumulating inside the morula, forming a cavity called the blastocele, and the embryo is now called a blastocyst. This transition marks the single most important cellular decision of your entire life. The cells of the blastocyst separate into two completely distinct lineages. The inner cell mass, also called the embryoblast, is a compact cluster of cells tucked against one pole of the blastocyst cavity. These cells, and only these cells, will form the actual baby. Every tissue of every organ in your body traces exclusively to this small group. The outer layer, the trophoblast, will never contribute a single cell to the baby itself. Its entire purpose is extraembryonic, forming the placenta, the chorion, and the syncytiotrophoblast that invades maternal endometrium to establish blood supply. Trophoblast dysfunction is the embryological root of preeclampsia and placenta accreta, high-stakes obstetric emergencies on every licensing exam.
So, how does a genetically identical cell at the center of the morula know it should become embryo, while an identical cell at the edge knows it should become placenta? The answer is positional information. The cell's physical coordinates within the embryo are its primary identity signal. This was proven elegantly in mouse morula experiments. Researchers labeled individual cells and transplanted them to different positions. A cell moved to the center consistently contributed to the inner cell mass. The same cell moved to the periphery consistently became trophoblast. The gene responsible for the inner cell mass fate is OCT4, while CDX2 drives trophoblast commitment, and these genes are mutually inhibitory. When you are surrounded by other cells on all sides, OCT4 wins. When you face the outside environment, CDX2 wins. This positional signaling logic, where your neighbors and your physical location determine your fate, repeats at every subsequent stage of embryonic development. It is the foundational principle behind morphogen gradients, organ axes, and pattern formation.
One of the most stunning revelations of modern developmental biology is this. The molecular signals that control development are not unique to humans. They are universally conserved across hundreds of millions of years of evolution. The proof? The PAX6 gene. PAX6 in humans is the master regulator of eye development. Mutations in PAX6 cause aniridia, a condition where the iris fails to form,
and are associated with Wilms tumor. Now here is the extraordinary part. When researchers inserted the human PAX6 gene into a developing fruit fly, a species separated from us by over 500 million years of evolution, it successfully triggered eye formation. Not a human eye, a compound fly eye, because the fly's cellular machinery interprets the universal build an eye command using its own evolutionary toolkit. The command is conserved. The hardware differs. This is why zebrafish, Drosophila, and C. elegans are such powerful models for human developmental disease. We share the same operating system.
This slide describes one of the most astonishing experiments in 20th century biology. Researchers took highly malignant teratocarcinoma cells, aggressive, rapidly dividing tumor cells, and combined them with a normal mouse morula. This chimeric embryo was then implanted into a surrogate mother. The result was a completely normal, healthy patchwork mouse. Half of its cells were derived directly from the tumor, yet no increased cancer risk, no malignant behavior. The tumor cells had been forced, by the overwhelming positional signals of the normal embryonic environment, to participate normally in development, differentiating, migrating, and functioning like any other cell. This experiment proved something revolutionary. Malignancy is not necessarily an irreversible state. It is a contextual state. The embryonic microenvironment's positional signals are powerful enough to override tumor cell behavior. This is now the foundational rationale behind differentiation therapy in oncology, using retinoic acid to force acute promyelocytic leukemia cells to mature rather than proliferate.
Look at this comparison table and let it sink in. Cancer cells divide rapidly and at high rates, exactly like embryonic cells. Cancer cells are highly migratory and invasive, exactly like embryonic cells during gastrulation and neural crest migration. Cancer cells are de-differentiating, losing their specialized identity, reverting toward the undifferentiated state of early embryonic cells. And cancer cells actively recruit new blood vessel formation through angiogenesis, just as the trophoblast does during placentation. Cancer is not random biological chaos. Cancer is a highly organized escape from adult regulatory constraints back to a fetal behavioral program. Understanding this reversion is why oncofetal antigens like AFP and CEA are tumor markers. Cancer cells re-express proteins normally only made during fetal life. And it is why embryological signaling pathways, Wnt, hedgehog, Notch, are among the hottest oncology drug targets today.
If we can crack the complete rulebook of embryonic positional signaling, 2 medical revolutions become possible. Frontier 1, oncology. Since cancer is a reversion to embryonic behavioral programs, understanding how positional signals control those programs gives us a blueprint to reverse malignancy. The teratocarcinoma mouse experiment was proof of concept. The clinical translation is differentiation therapy, already proven in APL with ATRA, and the emerging field of tumor microenvironment reprogramming. Frontier 2, regenerative medicine. If we can master the environmental signals that direct stem cell fate, the precise sequence of morphogens, growth factors, and mechanical cues, we can instruct pluripotent stem cells to generate any tissue on demand. Bioengineered kidneys, lab-grown cardiac patches, insulin-producing beta cell clusters for type 1 diabetes. Both frontiers share the same foundation, decoding the embryonic rulebook written in those first extraordinary days of human development.
Here is the final paradigm-shifting insight. Development does not end at birth. Birth is not a finish line. It is a checkpoint in a continuous biological program that runs from fertilization through growth, maturation, aging, and death. Aging itself is late-stage developmental biology. The same gene expression programs that build organs gradually lose their precision over decades. And at the extreme ends of this continuum, embryonic stem cells and cancer cells share a trait that is simultaneously terrifying and medically miraculous: effective cellular immortality, the capacity for unlimited self-renewal. Mastering the central enigma of embryology, how a single cell multiplies, differentiates, and deploys itself into a complete human being, is therefore the master key to understanding how life is built, how it breaks down in disease, and how regenerative medicine might one day repair it. This is why embryology is not just year one content. It is the blueprint of medicine itself.